Assalamu alaikum friends. I hope that you all will be fine. Friends, in this video I'm going to discuss about a very important topic in the protein metabolism, that is urea cycle. It is an important topic as a long answer question and as well as for short note. Okay, I will cover this topic under the headings of steps, regulation, energetics and disorders. Okay, so let's start our lecture. Okay, urea cycle. The main site for the process of the urea cycle, that is liver. This urea cycle process, it takes place inside liver. Okay. Now coming toward the other names of urea cycle. The other names of urea cycle include ornithine cycle, ornithine cycle and Krebs Henseleit cycle. These are the other names of urea cycle. Okay. Now coming toward the steps of the urea cycle. Steps. So first of all, the carbon dioxide and ammonia, they gets attached with each other. They gets condensed with each other, carbon dioxide and ammonia. And with the help of carbamyl phosphate synthase 1, carbamyl phosphate synthase 1, it forms carbamyl. It forms carbamyl phosphate. Okay, and here there is a utilization of two molecules of ATP, and there is a formation of two molecule of ADP and two molecule of inorganic phosphate. So basically there is a condensation, there is attachment of carbon dioxide and ammonia, and with the help of this enzyme, that is carbamyl phosphate synthase 1, there is a synthesis of carbamyl phosphate and there is a utilization of two molecule of ATP. Okay. In next reaction, in next reaction there is condensation of ornithine. There is condensation of ornithine. Okay, there is condensation of ornithine. Ornithine. And here there is also removal of inorganic phosphate. And with the help of ornithine transcarbamylase enzyme, ornithine transcarbamylase enzyme, there is a formation of ornithine. Sorry, citrulline. There is formation of citrulline. Okay, friends, I am repeating that there is a condensation of ornithine with the carbamyl phosphate and there is a removal of this inorganic phosphate with the help of this enzyme, ornithine transcarbamylase enzyme. There is a synthesis of the citrulline. This is the second step. Now coming toward the third step. In third step, the aspartate, it also gets attached with this citrulline. This aspartate, it gets attached with the citrulline, and with the help of arginosuccinate synthase enzyme, with the help of arginosuccinate synthase enzyme, and there is a formation of arginosuccinate. Okay, and also there is a utilization of ATP, and two highly energy phosphate bond, they are also utilized in this reaction. These two high energy phosphate groups, they released in the form of PPI, that is pyrophosphate, and leads to the formation of AMP and PPI, that is pyrophosphate, and there is a formation of organosuccinate. So this is the third step. Now coming toward the fourth step. In next reaction, with the help of arginosuccinate lyase enzyme, with the help of arginosuccinate lyase enzyme, there is a formation of arginine, and here there is a release of fumarate, and fumarate in next reaction, it is an intermediate of TCA cycle. It converts into malate, and malate converts into the oxaloacetic acid, and here with the help of malate dehydrogenase enzyme, this malate converts into oxaloacetic acid, and here NAD plus, it converts into NADH and H plus. Now, with the help of arginosuccinate lyase enzyme, there is a formation of arginine, and if this fumarate gets released in this reaction, and fumarate, it converts into the malate, and malate converts into oxaloacetic acid. In the last reaction, this arginine, it converts into the ornithine, and there is a incorporation of water, and there is a formation of urea, and there is a formation of this urea. Okay, with the help of an important enzyme, that is arginase enzyme. Okay, so friends, it is all about the steps of the urea cycle. I am repeating that there is a condensation of attachment of carbon dioxide and ammonia. With the help of carbamyl phosphate synthase 1, there is a formation of carbamyl phosphate. Two molecules of ATP are utilized in this reaction. In next step, with the help of this enzyme, ornithine transcarbamylase enzyme, there is attachment of ornithine with the carbamyl phosphate, and this phosphate group gets released in the form of PI, that is inorganic phosphate, and there is a formation of citrulline. In next reaction, there is attachment of aspartate with the citrulline, and with the help of arginosuccinate synthase enzyme, and two phosphate high energy bonds, they utilized, and there is a formation of this AMP PPI. PPI means pyrophosphate, and there is a formation of arginosuccinate. In next reaction, with the help of this lyase enzyme, there is a formation of arginine, and also with the help of arginase enzyme, again ornithine gets regenerates. So that's why the other name of urea cycle is ornithine cycle, because ornithine acts as a catalyst and also gets regenerates in this cycle. Okay, so these are the steps of the urea cycle. Now coming toward the this regulation process. Okay, मैं यहाँ पर एक पॉइंट स्किप कर गया हूँ कि यहाँ पर इस पर N-acetyl glutamate. This first reaction it also requires N-acetyl glutamate for the activation of this carbamyl phosphate synthase enzyme. It is very important. Now coming toward the regulation process. Regulation. The N-acetyl glutamate it is a positive allosteric activator of carbamyl phosphate synthase 1. This is the regulation. The N-acetyl glutamate it is a positive allosteric activator of carbamyl phosphate synthase 1. This is the regulation. Now coming to the energetic. Energetic. So friends, 2 ATPs they are utilizing here. 2 ATPs and 2 ATP they are utilizing in the form of PPI. So the total ATP is 4 ATP. The 4 ATPs they are utilizing in this urea cycle. 4 ATP. And this NADH and H plus, which is electron carrier molecule, this NADH and H plus in electron transport chain, it gives 2.5 ATP. So here 2.5 ATP. So the total amount of the ATP production that is 1.5 ATP. So net amount of ATP from this urea cycle that is 1.5 ATP. 2 ATP are utilizing here and 2 ATP are utilizing here, and the 4 ATPs total 4 ATPs are utilizing, and this one molecule of NADH and H plus in electron transport chain it gives 2.5 ATP. So the total amount of production of ATP that is 1.5 ATP. Now coming to the disorders. Disorders. Students, the deficiency of this enzyme, carbamyl phosphate synthase enzyme, it leads to hyperammonemia, hyperammonemia type 1. Due to deficiency of this enzyme, it leads to disorder that is hyperammonemia type 1. Now deficiency of this enzyme, ornithine transcarbamylase enzyme, the deficiency of this enzyme, it leads to disorder and that is hyperammonemia type 2. Hyperammonemia type 2. Okay, the deficiency of this enzyme it leads to disorder that is hyperammonemia type 1. The deficiency of this enzyme it leads to the hyperammonemia type 2. And the deficiency of this enzyme, arginosuccinate synthase enzyme, the deficiency of this enzyme it leads to disorder that is citrullinemia. Citrullinemia. The deficiency of this enzyme it leads to disorder that is citrullinemia. Okay. The deficiency of this lyase enzyme it leads to disorder and that is arginosuccinate aciduria. Arginosuccinate acid urea. The deficiency of this lyase enzyme it leads to the disorder that is arginosuccinate acid urea. And the deficiency of this arginase enzyme, it leads to disorder that is arginemia. arginemia. So these are the five disorder. The deficiency of this enzyme, carbamyl phosphate synthase 1, it leads to disorder hyperammonemia type 1. The deficiency of ornithine transcarbamylase enzyme, it leads to disorder that is hyperammonemia type 2. The deficiency of this arginosuccinate synthase enzyme, it leads to citrullinemia. The deficiency of arginosuccinate lyase enzyme, it leads to disorder that is arginosuccinate aciduria. And the deficiency of this arginase enzyme, it leads to disorder that is arginemia. There is another syndrome. These are the five disorder, and there is another syndrome that is called as HHH syndrome. This one is, this H means hyperammonemia. This H means hyperornithinemia, and this H means homocitrullinuria. This H means hyperammonemia. This H means hyperornithinemia, ornithinemia, and this H means homocitrullinuria. This collection of disorder, it is called a syndrome. Hyperammonemia, hyperornithinemia, and homocitrullinuria. This syndrome, it occurs due to defect in ornithine transporter 1 gene. It is due to defect in ornithine transporter 1 gene. So these are the five disorders and one syndrome, that is HHH syndrome. It is due to defect in ornithine transporter 1 gene. So friends, it is all about the urea cycle, their site. Okay, the first two reaction, they occur inside mitochondria, and the last three reaction, they occur inside cytosol, and the main site is liver. Okay, so friends, it is all about urea cycle, the site, steps, regulation, energetics, and disorders. If you have any query, you may ask in comment section, and keep watching, and thank you.